Hi Nerds, in this video we are going to talk about the physiology of the liver. We have a lot to talk about, so let's go ahead and get started. Alright guys, so the first place we're going to start off with is the liver functioning and metabolism. As you guys know, we've talked about all the metabolic pathways that we're going to kind of briefly cover in our metabolism playlist. So if you guys want to know about those pathways in more detail, go check that out in that playlist. What I want to do is, because the liver has so many pathways that it's involved in, I want to break it down into two individual pathways, the fed state and the fasting state. I think that's one of the best ways because it makes it a lot easier, it makes it more understandable. And again, we're not going to go through this in super detail. I just want to get the basic components of this and help us to understand what the liver can do. The liver is so amazing. I had a biochemistry professor once tell me that if you want to tell someone how much you love them, you say, "I love you with all my liver," because that sucker can do so many things. And by the time we're done with this video, you guys will truly see that. It is a magnificent organ. All right, so let's first off start off in the fed state with looking at carbohydrates. So as you know, the portal blood is going to be bringing a lot of nutrient-rich blood from the gastrointestinal tract. So one of those molecules that we're going to see a lot of being brought into our liver cells is glucose. You know, we bring glucose into our liver cells through GLUT transporters, particularly GLUT2 transporters. Now, what can we do with that glucose once we bring it into the cell? Well, I want you to remember that there is going to be at least 4 significant pathways, okay? One thing is, when we're in the fed state, we have high amounts of glucose being brought into our circulation. So because of that, there's going to be a very important hormone that's regulating all of these pathways that we're going to see in the fed state. That significant hormone that I want you guys to remember that is driving all of these pathways that we're going to see in the in the fed state is going to be insulin. So as you know, the pancreatic beta cells are going to secrete insulin, and insulin is going to control a lot of different pathways within the liver. Now, one of the things that happens is glucose is brought into the cell via the GLUT2 transporters. Once it's brought in, we can take that glucose and convert it into its storage form, okay? And that is going to be called glycogen. So that's one significant thing that we should know. So one pathway that we really should understand here is going to be glycogenesis. Let's do that in pink here. So this is going to be one pathway here that is really important, that it's going to be glycogenesis. Another pathway that's super important is going to be the hexose monophosphate shunt, or as you guys know it, the pentose phosphate pathway. So if you guys remember, glucose, when we took it through the pentose phosphate pathway, it led to the formation of ribose 5-phosphate. But that's not the significant thing that I want you guys to remember. There was particular molecules that were actually developed during this process. And those specific molecules that we generated is we took this molecule called NADP positive and generated what's called NADPHs. If you guys remember, this was important in fatty acid synthesis, it's important in nucleotide metabolism, it's important in antioxidant reactions, trying to regenerate glutathione into its reduced form. Really important. Okay, so that's the second pathway that I want you guys to understand, is it's going to also increase the activity of the pentose phosphate pathway. Another thing is insulin loves to catalyze catabolize glucose into the forms that we can utilize for energy. So what is that? We can take glucose, as you guys know, and if you know, we can break down glucose into 2 components. You form what's called GA3P, which is glyceraldehyde 3-phosphate, and you get another molecule called dihydroxyacetone phosphate. Now, as you guys know, these guys can isomerize into one another, but the significant thing is that these guys eventually end up forming pyruvate, 2 of them, okay? And then that pyruvate that we actually generate can be converted into what's called acetyl-CoA. And as you guys know, acetyl-CoA will go through the Krebs cycle, or the tricarboxylic acid cycle. We'll put TCA cycle for tricarboxylic acid cycle. And again, do remember it's also called the Krebs cycle. And then from this, we generate tons and tons of NADHs and FADH2s. And what these suckers do is they take these bad boys to the electron transport chain and they generate ATP. All right? So that is the significance that I want you guys to understand here. So One pathway is performing glycogen via glycogenesis. Two was performing ribose 5-phosphate. But the significant thing is that we're generating a lot of NADPHs via the pentose phosphate pathway. And the third thing is we're going to catalyze the oxidation of glucose into ATP formation. So that's a significant thing. Now here's the thing though, one more thing that we should realize. Glucose is in such high levels. So sometimes our liver will perform the glycogenesis, it'll form the pentose phosphate pathway, and it'll catalyze the oxidation of glucose into forming ATP. But guess what happens? Sometimes you form a lot of these acetyl-CoA molecules, a lot of these bad boys. Okay? And if you guys remember, we said that whenever there's excessively large amounts of acetyl-CoA, it inhibits the particular enzyme that converts citrate, right? You have what's called citrate and then isocitrate and then alpha-ketoglutarate. Well, the citrate is then, actually it's not going to be able to convert it into isocitrate. So if you guys remember, there was an enzyme that called Citrate Lyase that broke down into, so here look, there's a molecule called citrate, but because there's excessively high levels of acetyl-CoA, the citrate can't be converted into isocitrate. So what it does is, there's an enzyme called Citrate Lyase. And what that does is it breaks this guy down into two components. One is called oxaloacetate, OAA, and the other one is called acetyl-CoA. And if you guys remember, the acetyl-CoA was then converted into what's called malonyl-CoA, and that malonyl-CoA is what was made for fatty acids. Here, I won't be lazy, I'll actually write out here fatty acids. Fatty acids. So now we understand that there's actually going to be two things here. One is if there's excessively high levels of glucose, it'll lead to excessively high levels of acetyl-CoA. That'll have a lot of Krebs cycle activity, a lot of NADHs, a lot of FADH2s, a lot of ATP, that the Krebs cycle is going to be inhibited. Because of that, then citrate is converted into acetyl-CoA and then into malonyl-CoA and then into fatty acids. So this is the fourth thing that is important. It can also lead to fatty acid synthesis. And as you guys know, the liver is one of the organs that can generate glycerol. So we can also take this dihydroxyacetone phosphate and we can actually convert this bad boy into glycerol. And as you guys know, we can take the glycerol and the fatty acids, and guess what we can do with these bad boys? If we take glycerol and the fatty acids, we can make what's called triglycerides. I'm going to denote that as triacylglycerol, but it's the same thing as triglycerides. Okay, so again, that is the significant part here. What else is coming to the actual liver besides glucose? This is the big one, but it also is regulating amino acids. So you know amino acids are brought in. Let's do these bad boys in a baby blue. So now amino acids are brought into the cell, and then when they're brought into the cell, they're actually brought in via what's called a sodium-dependent cotransport. Okay, so sodium will be brought into the cell along with amino acids via cotransport. The main thing with the amino acids is these guys are going to be designed to form one really important thing. There is some other things that they're going to do, but I want you guys to remember the main thing within the liver is going to lead to the genesis of proteins. This is the really, really important thing that I want you guys to remember here, is amino acids are going to lead to the synthesis of proteins. Now, if there's excessively high levels of amino acids, and we actually have formed all of our proteins, sometimes what can happen is some of these amino acids can be converted into what's called acetyl-CoA. And that acetyl-CoA can do what? Can lead to the formation of citrate, because it's not going to go through the Krebs cycle right now. Some of it might go through, but we're going to assume that a lot of it's going into citrate, and then into acetyl-CoA, then to malonyl-CoA, and then into fatty acids. And then if we take those fatty acids and combine it with glycerol, what can we get? Triglycerides. So that's important to remember. Okay, so two things I want you guys to remember for this guy, for the amino acids. One is there's going to be primarily the synthesis of proteins. And the second thing is that if the amino acid levels are really, really high, we can take and convert it into acetyl-CoA or other keto acid intermediates and lead to the formation of fatty acids. And then the fatty acids can combine with the glycerol and form triglycerides. That leads us to the next guy. Okay, what about fats? Now you know the liver it actually is receiving a lot of its fat and cholesterol substances from these little structures called chylomicrons. I know you guys have we've talked about this primarily in the lipoprotein metabolism. All right, so now if you guys remember, we had these molecules called chylomicrons. These were important because they were taking a lot of the cholesterol. So what are they carrying with them? They're primarily carrying a lot of triglycerides. I'm going to put TAG. That's the big thing that they're carrying a lot of. And they're carrying a decent amount of cholesterol, as well as with other substances. But the significant thing is that we know that it's going to be bringing with the triglycerides. Now, when it takes it to the liver, there is particular enzymes that will take and break down the triglycerides into fatty acids and glycerol. So what we can get from this is we can get some free fatty acids. And what will happen is these free fatty acids will lead to, we can take and incorporate that into the formation of triglycerides. What else? Well, what the frick is this cholesterol going to be doing? We need to do something with this bad boy. So cholesterol in this time period, we can take this cholesterol and it has three fates. Okay, one fate is we're going to lead to the formation of cholesterol esters. So let's take this guy and let's say we make cholesterol esters. If you guys remember, this is the storage form of cholesterol. So this is the storage form. And we use a particular enzyme to do this called ACAT, acyl-CoA acetyltransferase. All we're doing is we're adding a different acyl group or acetyl group onto this cholesterol molecule. Another thing, and we'll talk about this in another video, is we use this cytochrome system, particularly CYP7E1. There's this enzyme called 7-alpha hydroxylase, and there's another one called 12-alpha hydroxylase, which are really important that take and add on hydroxyl groups onto cholesterol and convert these bad boys into bile acids. Again, we'll talk about this in more detail when we talk about the biliary system. And another thing that we can do with the cholesterol, let's bring it up one more, is we can take and incorporate these bad boys into something else. Let's say I take the cholesterol that I brought, right, from my chylomicrons. I take the triglycerides that I got from synthesizing it from glucose or from amino acids, and I take those triglycerides and this cholesterol that I have here. Say I take this cholesterol. And guess what I'm going to do with these bad boys? I'm going to combine them together and I'm going to secrete this out of the cell. Guess what this molecule here is called? Let's actually bring it. Let's keep it inside the cell here. So I'm going to take this triglycerides, I'm going to take the cholesterol, I'm going to combine these bad boys together and I'm going to get what's called VLDL molecules. And if you guys remember these VLDLs, we can put these out into the actual circulation and distribute a lot of the actual cholesterol and the triglycerides to the fat tissue, the muscle tissue. And if you guys remember, the VLDL will actually return. So it'll go to the adipose tissue, go to the muscle tissue, and then afterwards, it'll come back in the form of IDL, right? And some of that IDL will be taken up by the liver. Some of the IDL will then be converted into LDL, and we know that LDL will actually take that cholesterol to a lot of different peripheral tissue cells, and the remaining will be brought back to the liver. And then there's one more molecule, we actually secrete out HDL, and that HDL will take and pick up cholesterol from the peripheral tissues and bring a lot of that cholesterol back to the liver. So that's important to remember. Okay, so what are the big things that I want you to remember about the fat metabolism? One thing is I want you to remember that chylomicrons are bringing exogenous forms of triglycerides and cholesterol to the liver. The triglycerides, I want you guys to remember, we can have free fatty acids being formed. We can take and convert that back into triglycerides by combining it with glycerol. We also have the cholesterol coming from the chylomicrons. That can be converted into bile acids. We can take and convert it into the storage form of cholesterol, or we can incorporate it into lipoproteins, particularly VLDL. That VLDL will then do what? Go to the adipose tissue, go to the muscle tissue, drop it off, and convert it into IDL. IDL, some of that can come back to the liver and drop off the cholesterol. Some of it will become LDL. Then the LDL will take that cholesterol, drop it off into various tissues, which can use it for steroidogenesis. But unfortunately, as you guys know, some of this LDL can also deposit some of the cholesterol and lead to atherosclerosis if oxidized particles occur. And then we know that HDL is also another really important one that goes to these peripheral tissues and pulls a lot of that cholesterol off of the vessel walls, right? And then returns the remaining cholesterol back to the liver. So that's important to remember that. So that is the second thing that we should also know here. Okay, now we've gotten a really good understanding of what's happening in the actual fed state with glucose. We know what's happening in the basis of amino acids, and we know the basis that is happening with the fat molecules. Now what I want to do is I want to take it a next step, and I want to see how this is actually, the metabolism occurring in the liver during the fasting state. All right, so now what we're going to do is we're going to look at the metabolism that is occurring in the liver in the fasting state. So now we're looking at the opposite here. We're looking at, it's been a while since we've eaten, our glucose levels in the blood are starting to drop, and there's a particular hormone that's being released in response to these low blood glucose levels, and that hormone is going to be glucagon. Okay, glucagon is, if you guys remember, released from the pancreatic alpha cells. There's other hormones that are also released. You have cortisol that could be acting in this time period. You have another one, that being growth hormone. Other ones could be like epinephrine. There's even norepinephrine. A bunch of these guys. Even thyroid hormone, if you want to throw that bad boy in there too. T3 and T4. So a lot of these guys are going to be driving a lot of the fasting state processes. So what's happening here in the fasting state? Here's what I want you guys to remember. Let's go back and let's look at this with respect to glucose. Let's look at it with amino acids, and let's look at this with respect to fats. So the first thing is now our blood glucose levels are low. That's the stimulus here. What was the stimulus here? The stimulus was low blood glucose levels. Okay? Our goal in the fasting state is to increase the blood glucose levels. So one thing that we're going to want to do is we're going to want to be able to break down that reserve of glycogen into glucose and put that into the blood. So that's one of the definite things that we're going to want to do here. So let's say we take this glycogen that we had stored here, and now what I'm going to do is I'm going to break this down into glucose. So this is called glycogenolysis. So this is one important pathway here. This is one big thing here. Okay, glycogenolysis. What else? Okay, well, we did glycogenolysis. Let's do something else. Let's do the opposite of glycolysis. Let's do gluconeogenesis. So you know why this is so cool? If we take amino acids, so we can take amino acids and we can convert these amino acids via what's called transamination. We've talked a lot about this. We can convert this into glucose. So that's one really important thing to remember is we can take these amino acids and we can undergo what's called a transamination process where we convert into different keto acids, particularly pyruvate, and that pyruvate could lead to the formation of glucose. So that's one, another pathway that can happen. Another thing is, glycerol, glycerol is actually a product of lipolysis. So what we can do here is we can take the glycerol that we can get from lipolysis, we can take that glycerol that we can get from the lipolysis that is occurring in the adipose tissue or in the muscle tissue or even liver tissue, and we can take that glycerol and we can convert that into glucose. So that's another pathway that is occurring here. So that's the third thing. Another situation is your muscles, they release a lot of lactic acid. And a lot of that lactic acid, as it builds up, we can actually take that lactic acid and use it for good. We can take that lactic acid and convert that into glucose. That is the fourth thing. And all of these processes are going to be under what's called the gluconeogenesis pathway, right? So gluconeogenesis is taking non-carbohydrate molecules and converting them into glucose. So we take amino acids, glycerol, lactic acid. If you want to throw one more in there, there's also odd-chain fatty acids. We can do that. But they have a minor contribution to the blood glucose levels. Okay, so that's the big thing there. Now, something else that we should remember. When we take these amino acids and we try to convert them into glucose, the process of amino acid catabolism can push out something. So You know, during that process, when we break down, we take the amino acids and we try to convert it into glucose, we generate what's called glutamate. Okay, you know, you get this usually as a product of transamination. That glutamate is then going to do something in the liver. You know, glutamate, it can actually rip off an ammonia molecule off of it. There's a glutamate dehydrogenase enzyme that works in this pathway. And then what will happen is that glutamate, when it rips off the actual amine group, it converts this guy into alpha keto glutarate, which is a Krebs cycle intermediate, right? Well, guess what happens with that ammonia? This is a super toxic molecule. Because it is so toxic, what we can do is we can send this bad boy through a cyclic pathway called the urea cycle. The urea cycle. And in the urea cycle, we use particular enzymes to take and push out of this a molecule called urea. And then you know urea is less toxic, and it's actually, we're able to send this to a particular organ in the body. You know the kidneys? The kidneys can actually take up that urea and excrete that urea, right? So look, we're gonna take and pee that sucker out. So now what we're gonna see in the urine is we might see a lot of urea. Okay, that's one important thing. This accounts for the ammonia going through the urea cycle and producing urea, accounts for 95% of the ammonia metabolism. But guess what? There's another guy. You see this ammonia molecule, he can go right back and bind onto glutamate. So he can take and he can bind right onto glutamate. And when these two guys bind together, let's actually do this like this. These guys, when formed, they can lead to a molecule called glutamine. This is an important one to remember because there is an enzyme that I should mention that drives this process, and this is called glutamine synthetase. So glutamine synthetase is the enzyme driving this process, and it's converting the glutamate to glutamine, which is going to pick up that ammonia molecule. And guess what we can do to the glutamine? We can send this bad boy to the kidneys and excrete out the glutamine into the urine. And this accounts for 5%. So the formation of glutamine accounts for 5% of the ammonia handling there, okay? So that's one important thing that we should remember. As a result of amino acid metabolism, particularly in the fasting state, when the amino acids undergo the transamination process and are converted into glucose via gluconeogenesis, or we can take the amino acids you'll see and not just convert into glucose, but we can utilize it for energy. During that process, we generate what's called glutamate. And then glutamate pulls off an ammonia molecule. We get an ammonia molecule pulled off of it, and that's called oxidative deamination that leads to the formation of alpha ketoglutarate. That ammonia molecule has two pathways. One is it can be converted to glutamine, which can be urinated out, only accounts for 5%, or it can go through the urea cycle and form urea and be PP'd out. Reason why this is important, because if ammonia is not actually undergoing this cycling process, ammonia levels can build up in the blood. Why is that dangerous? Because it can lead to a lot of problems within your central nervous system, particularly what's called hepatic encephalopathy, which can happen whenever there is certain situations like hepatitis or cirrhosis or Wilson's disease or maybe hemochromatosis, infections of the liver, whatever it might be that's damaging the liver and decreasing its function. It can cause problems with this ammonia handling. Okay? So far we know that we're doing glycogenolysis, gluconeogenesis, glutamine synthesis, the urea cycle. Something else that we should understand, and I already said it already, is some of these amino acids, they don't just have to undergo this gluconeogenesis. Some of the amino acids can actually be converted into, it might actually be converted into, for example, let's say I take these amino acids and I send them into the TCA cycle. Okay, the TCA cycle. And then from here, we can take these guys, send it to the electron transport chain, and lead to the formation of ATP if necessary. Okay, so that's another thing that we should understand here, is that we can take amino acids if we need to, and we can send them into the tricarboxylic acid cycle and generate some energy from those molecules as well. So they don't always have to go into the formation of glucose. Now, what about the fat? This is a really, really important one. So if we take the triglycerides, because our liver can actually have, it stores, we can have triglycerides. Okay, so let's say TAG. The liver can break down the triglycerides into two components. One is obviously called glycerol, and we know where that molecule went. We know that it went to form glucose. Another thing is the triglycerides can lead to the formation of what's called fatty acids. Now, the fatty acids can undergo this process where they have what's called beta oxidation. And that beta oxidation will take the fatty acids and convert it into what's called acetyl-CoA. Okay? Now this acetyl-CoA can take and go through the Krebs cycle, right, or the TCA cycle. And then from here, we might take some of the NADHs and the FADH2s, send them to the electron transport chain, and make some ATP. But guess what? Because sometimes there can be a lot of fatty acid metabolism, and again, what is this process here called? It's called beta oxidation. Sometimes in the liver, if we have a fasting state for a long period of time, the acetyl-CoA molecules can build up. Why is that important? Because these acetyl-CoA molecules, if they build up, they can be converted into these special little molecules called ketone bodies. Okay? And these ketone bodies, there's actually of two types, two different flavors. One is called acetoacetate, and the other one is called beta hydroxybutyrate. And technically there is another one. If you really want to be specific, there's even acetone. But really this one is going to be breathed out or exhaled. Okay? So that is another thing that we should remember can happen in the fasting state. So far, what do we have? Glycogenolysis, gluconeogenesis, we have glutamine synthesis, we have the urea cycle happening, we even have lipolysis that can be occurring here. So we can have lipolysis, that's the 7th part, and we can have beta oxidation, and if there's excessively high levels of acetyl-CoA, we can take and convert that into ketone bodies. So that is the significance that we should understand here with the liver that is happening, what's happening in the fasting state. Okay, so now what I want to do is we got a good chunk of the metabolism down. Again, if you guys really want a more in-detail look at this stuff, we covered this in our metabolism playlist, so go there, check out all those videos. I just wanted to get a basic little thing here for how the liver is involved in metabolism. All right, guys, so now we're going to talk about, actually, let me flip these guys here. I think it's better if we talk about protein factory first, because I think it's going to make more sense. It's going to flow better anyway here. So let's go ahead and switch these, and let's make this one here. Let's make this the protein factory. Let's cover this as the protein factory, because the liver has so many proteins that it can synthesize. You guys will see it's unbelievable. And then we'll talk about storage last. Okay? So protein factory. Obviously you know that the liver has the ability to make tons and tons and tons of proteins. Now, here's the thing. What I want us to look at this in is I want us to look at this in a bunch of different ways. All right, so this guy makes tons and tons and tons of protein. So what are some of the proteins here? Out of all of these, guys, don't forget this one. This is the most significant protein that the liver makes, and this is called albumin. You guys already know that albumin accounts for like 60% of your plasma proteins. Now, what is the function of albumin? Albumin maintains your osmotic pressure. So for example, let's say I had a capillary here. So here is a little capillary. You know that in the capillary there's hydrostatic pressure, pressure that pushes things out of the actual capillary, like different solutes and water and electrolytes. And then there's another pressure which tries to keep the water inside of the actual capillary. Albumin is playing a role in this. And the reason why that's important is imagine here's the tissue cells, right? So here's a couple tissue cells. In between the blood and the tissues is the interstitial fluid, right? Now, let's imagine here for a second that your albumin levels were low. What would happen? A lot of the water in this area would accumulate a lot. And then that would lead to swelling of the interstitium. That's called edema. That's why albumin is so, so important, and we should really understand this guy. Also, when people have a long-term or severe injury to the liver, like due to hepatitis or cirrhosis, whatever the etiology might be, one of the long-term signs that there is liver damage is the albumin production starts decreasing. As well as, you'll see in other proteins that they make is they make a lot of clotting factors. So what's called their prothrombin time, the time it takes for them to clot basically, that's also a lot longer, okay? So that's the really important protein that I want you guys to remember here. Other ones, it makes a bunch of different proteins, and these guys are called your globulins. There's alpha and there's beta globulins. Now, out of these guys are your transport proteins. They're the ones that transport a lot of different lipid-soluble substances, like maybe steroid hormones or steroid-based substances, or things that like trace minerals like copper and iron and stuff like that, okay? So for example, just to name a few of these guys, let's say I have one, I already mentioned copper. There's a protein that transports that. It's called ceruloplasmin. Okay, super, super important. It transports copper. Reason why this is important is you know that there is a disease called Wilson's disease, pretty rare, but what happens is it's copper overload disorder. There's a protein called ATP7B, and it's what's responsible for taking the copper and combining it onto the ceruloplasmin. If that protein is actually defective, then the copper levels will actually start increasing and lead to a lot of free radical reactions. So that guy's super important that we should understand. Also remember that the copper is also secreted into our biliary system too. Other ones, we also mentioned iron. There's another protein called transferrin. And transferrin is responsible for transporting iron. He takes the iron and delivers it to the various different tissues. One is it could be the bone marrow because that's needed for erythropoiesis, and it takes it to the liver. And the liver will actually store that iron in the form of ferritin, or if we have a lot of ferritin, that can polymerize and form hemosiderin. Okay? Other ones, haptoglobin. Haptoglobin. Sometimes, you know, when there is hemolysis of our red blood cells, the breaking of the red blood cells, hemoglobin can be released. And some of that hemoglobin, we need to bind it onto haptoglobin. Okay, because hemoglobin is actually pretty toxic inside of our actual blood on its own. So that's important too. Other ones, let's say that we have, we have what's called retinol binding protein. So retinol binding protein, this guy's super important. He actually, you'll see later when we talk about the storage function of the liver, the retinol binding protein actually binds onto retinol and transports the retinol to various different tissues. What is one of the big tissues that retinol is needed in? In the eyes. It's needed for the actual dim light vision, right? So that's important. It's also utilized in our skin. It also transports different types of steroid hormones. For example, cortisol. Cortisol is actually transported via what's called transcortin. And then thyroid hormone. Thyroid hormone is transported in the form of what's called thyroxine binding globulins. So tons and tons of proteins. I'm not going to keep going through all of these bad boys. But I just want you guys to get the idea that the alpha and beta globulins are designed to be able to act as carrier proteins, to transport substances that are lipid soluble, they're hydrophobic in other words, or that can be super dangerous and can lead to a lot of free radicals, specifically the iron and the copper. These guys can lead to a lot of dangerous reactions, particularly if you guys know, we'll talk about it later, the Fenton reactions. Pretty dangerous stuff. Okay, so they're transporting steroid-based substances or ions or metals, okay? Well, some other proteins that the liver makes besides these bad boys. It also is responsible for making different types of clotting proteins and anti-clotting proteins. So, for example, let's say I have clotting proteins. You guys know a lot of these. There's so many of them, right? So, but here's the ones that I want you guys to remember. The big ones. Factor 2, okay? Factor 7. Factor 9. Factor 10. protein C, and protein S. The reason why I want you guys to remember these is these suckers right here are vitamin K dependent. So in order for these proteins to become active, they need to have vitamin K. Okay, so that's some really, really important ones. So it makes a lot of different clotting proteins. Another one that's really important here in the clotting cascade is it also makes what's called fibrinogen. Okay, fibrinogen, which is actually factor 1. That's important for the fibrin mesh. As you guys remember, fibrinogen was acted on by factor. There's actually thrombin or factor 2. And what that did is that converted fibrinogen into fibrin, and then fibrin was responsible for forming that fibrin mesh that anchored down that platelet plug. And if you remember, factor 13 actually takes and cross-links the fibrin strands to really anchor down that fibrin mesh. So that's really important. As a side, to oppose the clotting, it also likes to keep a nice little balance. It also makes these anti-clotting proteins. So if here, let's word it like that, anti-clotting proteins. So what are some of these bad boys that it makes? It makes what's called, you guys should know this one, plasminogen. Plasminogen, super important one. If you guys remember, plasminogen is acted on by an enzyme called TPA. Right? Whenever someone has an acute thrombus or something like that, whenever they might have a clot, we give them TPA. Why? Because TPA converts plasminogen into plasmin. And what was plasmin's job? Remember, we represented this as like a nice little Pac-Man-like enzyme. And what was it breaking down? It was breaking down, look at this, the fibrin. And it was spitting it out in the form of fibrinogen. And sometimes you might even see it in the form of what's called D-dimers. Sometimes that's why somebody has an elevated D-dimer, they might have actually had some type of clot, right? So that's really important. He makes plasminogen. What's another really important one that he makes? Another really important guy is called antithrombin 3. If you guys remember, antithrombin 3 binds with heparin sulfate on top of our endothelium. And antithrombin 3 was responsible for degrading particular types of clotting factors. And what were some of those? Out of all of these, if you guys remember, just remember that it inhibits or deactivates factor 2, which is thrombin. It also deactivates factor 9, and it also deactivates factor 10. So that was some of the things that you guys should remember here. Okay, so we got clotting proteins, anti-clotting proteins. What are some of the other ones? Here's what I love about this guy. He also can make hormones. So what are some of these hormones? This is what's super interesting. Most people don't know, most people forget about this when they think about the liver. Another thing's really interesting about the liver, I know I go off on tangents a little bit, but you know the liver is actually the site of red blood cell production in the fetus. So it actually starts off in the primitive yolk sac, and then the liver carries out the erythropoietic process till about the 32nd week of gestation when the bone marrow takes over. But there's another thing that it also controls. It actually makes a particular hormone called thrombopoietin. And thrombopoietin, if you guys remember, we talked about it very, very briefly. There's what's called the myeloid stem cell, if you guys remember that, right? And what it does is thrombopoietin actually leads to the platelet synthesis. So platelet formation. If you guys remember, we said that thrombopoietin will take a pluripotent stem cell. Well, you have what's called pluripotent stem cell or hemocytoblast, converts it into a myeloid stem cell. And then thrombopoietin will stimulate that myeloid stem cell to turn into a megakaryoblast. Then it goes to a promegakaryocyte, and then a megakaryocyte. And then the megakaryocyte starts shedding platelets. And then these platelets are important for actually helping to form blood clots, right? For plugging different holes inside of the blood vessels. Another hormone is called Insulin-Like growth factor 1. We talked about this in the growth hormone video, okay? And what does this guy do? If you guys remember, it acted on muscles, it acted on bones, it acted on cartilage, and it increased the protein synthesis in these guys. So it helped to be able to increase the muscle size and muscle density. It helped to be able to increase the endochondral ossification. It also helped to increase interstitial growth and collagen type 1 production. So that's an important one. What's another one? This guy's so darn cool. Angiotensinogen. And if you guys remember, angiotensinogen is acted on by renin, which is secreted by the JG cells in the kidney. That converts angiotensinogen into angiotensin 1. And then angiotensin 1 is acted on by ACE, angiotensin converting enzyme in the lungs. And eventually, that's converted into angiotensin 2. And if you guys remember, angiotensin 2 is super important. It involves an ADH release, aldosterone release, vasoconstriction of our blood vessels, all designed to increase the blood pressure. So we know it has albumin, alpha-beta globulins, clotting proteins, anti-clotting proteins, hormones. What's another really important one that I want to finish off with here? Let's do one more really big one, and that's immune, okay? It also makes a lot of different immune proteins. Now, out of these, there's at least 2 that you should remember, okay? Let's just do 2. So out of these, one is the complement proteins. We talked about this in our immunology playlist, right? There's complement proteins C1 to C9, and they're involved in the alternative pathway, the classical pathway, and even they're also involved in the lectin pathway. What was the significance? So the big significance with these guys. They're involved in what's called opsonization. What does that mean? Okay, let's imagine here I have a piece of bread. Ugh. But then you take some Nutella. Rub that all across that bad boy. What does it do? It makes it a lot tastier. That's what these complement proteins are doing. They're increasing the tastiness of the bacteria or pathogens, which enhances the phagocytosis process. And you also remember that they can act as chemotactic agents, C3A and C5A, okay? One more is C-reactive peptide. Please remember this one. C-reactive peptide. So C-reactive peptide is important because this guy is when, let's say you take a blood panel, you test their blood, and you see that they have elevated C-reactive peptide levels. What does that mean? That means that the person has active inflammation. It's a sign of that, right? There's a hormone, interleukin, called interleukin-6, as well as interleukin-1 and tumor necrotic factor alpha that can be released whenever there's inflammation by macrophages. And it stimulates the liver to produce C-reactive peptide, and that's actually an inflammation marker. But what C-reactive peptides do is they act as opsonins. So they actually activate the complement system, and that is important. So if you can see here, look at this. This is just unbelievable what the actual liver can do. And this doesn't include all of them. That's what makes it even more amazing. Okay, so this is just a few of the proteins that the liver is making that I want us to kind of just get a basic view of. Oh, one more that I completely forgot about. Shouldn't forget this one. Is it's also responsible for making these things called apoproteins. Apoproteins. We talked about these a lot in lipid metabolism, the lipoprotein metabolism. And if you guys remember, it was ApoA, ApoB, ApoC, and ApoE. Obviously, we talked a lot about ApoA1, B100, B48, C2, and ApoE. So it synthesizes a lot of those apoproteins, which is significant in our lipoprotein metabolism. Okay, so that covers the gist of the protein factory. Let's come over here and let's hit it home with the storage function. All right, so to finish it all up, guys, we're going to talk about the storage function of the liver. Okay, so here's what I want you guys to remember. It stores fat-soluble vitamins, primarily A, D, E, and K, as well as vitamin B12, okay? And we'll talk about its storage of trace minerals like copper and iron, okay? So let's start off first with the vitamins. So obviously, all of these vitamins. So here, let's imagine here you have what's called your chylomicrons. And the chylomicrons, if you guys remember, they were transporting triglycerides, cholesterol, phospholipids, as well as vitamins A, D, E, and K. Now, these chylomicrons, you know, they go through the lymphatic system and into the circulation, and then they get taken to the liver. Now let's see what it does with each one of these guys. I don't want to go crazy over all of them. I want to focus on two primarily. So let's do those two important ones that I want to really focus on. That's vitamin D and vitamin K. So a lot of the vitamin D can be brought via the diet. But here's the other one that I want to talk about. A significant amount of vitamin D is actually coming from our skin. You know, our skin has a cholesterol molecule in it called 7-dehydrocholesterol. So what is this molecule called? It's called 7-dehydrocholesterol. Holy frick, that's a long name, right? So when sunlight, let's say photons, look at this. Here's some photons. Some photons are hitting the skin. It actually can take the 7-dehydrocholesterol and convert it into another molecule. And what it does is it converts into what's called cholecalciferol. I'm not going to spell it out, that's too crazy. CCF. Now, this cholecalciferol will then go to the liver. Okay, now the liver's job in this is it takes, let's say here's the liver. Here's the liver, here's a little baby liver. Here's your falciform ligament. It has an enzyme called 25-hydroxylase. So we're just going to put here this guy right here. There's an enzyme called 25-hydroxylase. And what that does, it puts a hydroxyl on the 25th carbon of this cholesterol derivative. And this is now going to be called 25-hydroxycholecalciferol. Now from here, it'll then go to another organ. It'll go to your kidneys. Okay, so now let's just draw in a little baby kidney. And let's get this little guy over here. Little baby kidney. Okay, now the little baby kidney has a particular enzyme in it, and this is called 1-hydroxylase. 1-hydroxylase. What that 1-hydroxylase does is, what do you think it does? It puts a hydroxyl group on the one carbon of this cholecalciferol, which is this cholesterol derivative, and converts it into 1,25-dihydroxycholecalciferol. Holy crap. Now, what is this guy? This is the active form of the vitamin D. That's the active form. Now, what this does is this 1,25-dihydroxycholecalciferol is he goes to your GI tract. So for example, he'll come here to your GI tract, and when he comes to your GI tract, it increases your calcium absorption. It activates particular genes that increase the synthesis of calcium binding proteins, and that's going to lead to increased calcium absorption. Pretty cool, huh? All right. So that's the one there. So this one right here is specifically for your vitamin D. Okay? So that's its function there. What's another thing that it can do? Another thing is we said we were going to focus on vitamin K. Okay, so what is vitamin K here? Let's actually take here this baby blue here. So now, again, here's going to be the next one, vitamin K. Well, vitamin K, here's what I want you guys to remember. Super, super important, and what we already talked about in certain clotting proteins. So what happens is, here's the cool thing with the vitamin K. Vitamin K comes in two forms. There's what's called the oxidized form, which is usually in the form of an epoxide. And then there's another one called the vitamin K that's in the reduced form. So now this is in the reduced form. And then these guys can go back and forth. So we can take this vitamin K in the oxidized form, convert it into vitamin K in the reduced form. Now, there's an enzyme here. Let's do this guy here in red. This is called, that converts the oxidized form into the reduced form. And this is called vitamin K epoxide reductase. Now, why is that important? You know, there's a drug called warfarin. They use it in trying to treat patients who have, increased incidences of clotting or maybe even atrial fibrillation. They use this as a blood thinner because what it does is it inhibits the vitamin K epoxide reductase. If that happens, vitamin K in the oxidized form can't go to vitamin K in the reduced form. Why is that important? I'm glad you asked, guys. Because guess what happens? There's proteins, factor 2, factor 7, factor 9, factor 10, protein C, and protein S, so protein C and protein S, and if you really want, there's another one called protein Z. But all of these guys, they're in this inactive form, so inactive. When they interact with the vitamin K in the reduced form through a specific enzyme here called a carboxylase enzyme, it converts these guys into their active form. So that's important here. And again, what is the enzyme that's triggering this process to go from the reduced back to the oxidized? This is called a carboxylase enzyme. Okay, so that is the significance there with the vitamin K. Now, the vitamin A, what's its significance here? Well, the vitamin A is pretty darn cool as well, but there's not a lot that we really need to talk about with this one. So for example here, let's say I take the next one, the vitamin A. The vitamin A, when we absorb it across our GIT and the form within the chylomicrons, it's usually coming in the form of what's called retinal esters. What happens is we take this bad boy to our liver, okay? And the liver, let's draw another little baby liver, okay? What it does is it has particular enzymes that convert the retinal esters into retinol. And then another thing the liver does is it produces a protein that will actually bind onto the retinol. Look at this. Here we're going to have a protein that binds onto. We already mentioned this protein. It's called retinol binding protein. Look at that. And then guess what can happen here? This retinol, when it's bound to the retinol binding protein, we can take it to different peripheral tissues. What are some of the tissues? One is it can go to the skin, and the other one is we could take it to, the rods within inside of the eyes, like particularly in the retina, right? Because it's important for our dim vision. So that's one thing. Another thing is we can take that retinol, and instead of binding it onto the retinol binding protein, guess what we can do with that retinol? We can store it. So it can be stored in these special little cells located in the space of Dissy. And these are little cells here. Guess what these little buggers are? These are called your stellate cells. And your stellate cells are important because they can take that retinol and they can store it. So that would be important within the storage of the actual vitamin A, and then it can be released out into the circulation, bind onto the retinol binding protein, and then be utilized to different various tissues, right? So that's the significance there. The next one is vitamin E. Now, vitamin E is also important, but he is really, really specific in the antioxidant function. So again, we take it through the chylomicrons, take it to the liver, and the vitamin E, okay, it's actually carried via the chylomicrons. When we take it to the liver, what the liver actually does with it is it'll take the vitamin E and incorporate it into what's called your VLDLs. So it'll incorporate it into the VLDLs. And also, the VLDLs, we know that some of it actually is converted into IDLs, some of it's converting into LDLs. So what's important with this is it has the antioxidant function. Okay, because you know what happens when the LDL particles can undergo oxidation, it can lead to a atherogenesis. So guess what this vitamin E is for? It's specifically carried within a lot of these lipoproteins to prevent the oxidized particles from actually causing a lot of the atherogenic effects. So its antioxidant function can be designed to try to decrease atherosclerosis or atherogenesis. So that's pretty darn cool if you think about it. Okay. Now for the last two buggers here, let's say we have to talk about the last one here. Oh, and one more, vitamin B12. Again, vitamin B12, not crazy a lot of stuff to talk about with this one. Just remember that vitamin B12, we can actually take this and it actually is transported to the liver. So let's imagine here there's a transport to the liver, and when it's transported to the liver, it's actually transported by a specific protein produced by the liver. So look at this. There's a protein here made by the liver, and this protein is called transcobalamin. And if you want to be specific, it's actually transcobalamin type 2. And it binds onto the vitamin B12 and takes it to the liver. And then the liver can actually store that vitamin B12 in the form of transcobalamin 1, or it can secrete that out, right? So we can take that vitamin B12 and we can take it to various tissues. What's one tissue that is really needed for the vitamin B12? The bone marrow. So we can take this to the red bone marrow, right? And the red bone marrow needs this. And the reason why the red bone marrow needs this is it's a precursor or a necessity for erythropoiesis. What else? You know, vitamin B12 is also really important for DNA synthesis. It's involved in the folic acid pathway, right? So that's important too. Okay, so we covered the basic of the vitamins. Now, the next thing that I really want to talk about is the copper and the iron. So let's say here I have, we've already talked a lot about this in the individual videos, but let's say here's an enterocyte. And you know that the iron is brought in to the enterocyte via specific proteins like DMT1, divalent metal transporter. And then from here, we can actually push it out through particular proteins located on the basolateral membrane. This is the really important one called ferroportin. If you guys remember, it's very important, or ferroimportant. And then what happens is the iron there is bound to a particular protein. And what is that protein here that binds onto it? That protein is called transferrin. And then that transferrin, if you guys remember, is going to take the iron to the liver. And the liver is going to take up a lot of this. So now it's going to take up the iron. And what it's going to do with the iron is it's going to store it, right? So we can take this iron. Let's say here's the iron. We can take it inside of these liver cells, and we can bind it to what's called ferritin. Okay, actually it's originally apoferritin. When it binds into the iron, it becomes ferritin. And if there's a lot of ferritin molecules, they can be converted into hemosiderin. This is important because whenever there's high levels of this hemosiderin or high levels of the iron, the liver makes a protein called hepcidin. And then hepcidin, if you guys remember, actually is responsible for regulating the ferroportin. So if there's high levels of iron, the actual liver will produce a protein called hepcidin. Hepcidin will inhibit the ferroportin, which will inhibit the iron absorption. Why is this important? Because if some people are not able to release the hepcidin, if there's a deficiency or a mutation, it can produce a disease referred to as hemochromatosis. Okay? Why is this super dangerous? Why is iron so dangerous if it's built up in a lot of levels, high levels? Iron can actually react with hydrogen peroxide inside of our cells. When it does that, it converts the iron from the +2 state to the +3 state, and it forms this really nasty molecule here called a hydroxyl radical. And this guy can cause a lot of damage. Okay, what kind of damage? It can damage our proteins, it can damage our DNA, it can damage our cell membranes. So super, super nasty free radical. And this can actually, the dangerous thing here is that sometimes this guy right here can go right back to the iron 2+ by reacting with hydrogen peroxide. This is really dangerous because this is called your Fenton reactions. And this is what can be really dangerous because it can actually damage the liver cells, and some of the iron can leak out into the blood and go to various tissues and damage those tissues as well. Okay, and the last one that I want to talk about is copper. So let's say we take the copper here. Let's do let's actually do this maroon. All right, so let's say here we have an enterocyte here. And again, we're going to take up our copper. Now here's what you got to remember. There is these little transporters there, divalent metal transporters that take up the copper. Now, the copper, when we take this bad boy up, we actually have the copper bound to another protein here. Let's say here's this protein. This one transports it to the liver. And this protein that binds onto it is actually called albumin. And then what will happen is this copper will be transported to the liver. Now, what happens with this copper? Let's say we take this copper out here. This copper here is super cool because what happens is he's going to bind on to, there's going to be a kind of like a protein shuttle, if you will. Let's take this guy here and let's draw him as ATP7B. Okay, so he's a super important protein that binds onto the copper and takes the copper and puts him into two different destinations. One is he can take the copper and transport it and actually excrete it out into the bile. Okay, so it can be excreted into the bile. There are specific transporters like MDR1. We'll talk about that, multi-drug resistant transporters, that it can excrete out the different types of organic cations. Okay. Another thing is, and this is just to maintain, we don't want to have copper build up inside of our liver cells because it's toxic. It can actually lead to free radical reactions. Some of it actually is going to bind onto the ATP7B, and it's going to be bound to, it's going to be transported, and there's another protein inside of our cell called apoceruloplasmin. When it reacts here, the ATP7B transfers the copper onto the apoceruloplasmin and converts this bad boy into ceruloplasmin. Now, here's the thing. In a crazy disease called Wilson's disease, there's damage, or I shouldn't say damage, there's a defect or mutation in which this ATP7B isn't produced. If that's the case, the copper can't be excreted into the bile, and the copper can't be bound onto a ceruloplasmin. So the free copper starts rising inside of the actual liver cells. Why is this bad? Same thing is that copper, what it can do is it can react with hydrogen peroxide. And when it reacts with the hydrogen peroxide, guess what it does? It takes and converts that into copper 2+. And this super dangerous molecule, let's put him in pink here, the hydroxyl radical. And again, what can this guy do? He can lead to a lot of oxidative stress. He can damage your DNA. He can damage your proteins. He can damage the cell membrane via lipid peroxidation. And so it's a really, really nasty little thing here. So again, this is really important that we should understand how the liver is actually storing all of these different types of fat-soluble vitamins or water-soluble vitamin, storing iron and storing copper, because if for just some reason there's a mutation in this gene, it can produce Wilson's disease. And Wilson's disease, you actually know how you can see that, one of the diagnostic signs? Copper can actually get put into a specific membrane inside of the cornea. You know, there's what's called the Descemet's membrane. And when the copper accumulates there, it produces these like brown pigmented rings called Kaiser-Fleischer rings. And that can be one of the diagnostic signs that there could be a copper accumulation, and also can start really damaging your central nervous system. If you guys watched this whole video, can't thank you guys enough. I really appreciate it. If you guys like this video, please, I'm begging you, hit that like button, comment down in the comment section, and please subscribe. Also, if you guys get a chance, go check out our Facebook, our Instagram, and even our Patreon account. All right, Ninja Nerds, as always, until next time.